So now this next slide is we're going to get Dr. Conrad to comment on too, because it gets into the specifics of what we're going to be looking at when we come back with results. Hopefully towards the end of the summer. Can you take us through this? It's a little complicated, but I think you can break it down between the both of you guys.
Yeah, so I'll briefly. So this is essentially an example of what we will see from this trial from histology, so pure biology. And there are 4 panels here. On the left, this is control. This is an untreated tumor. And then on the right, the next panel, panel 2, is what would happen if you treat something with an immunotherapy, in this case, interleukin-2, and the immune therapy is ineffective. And what you're seeing there is it's all clustered at the edge of the tumor. It actually doesn't penetrate into the tumor. So the tumor to the right is free of the effect that we're trying to achieve. And we see this in cancers very often in the human setting, that person on immunotherapy has immune cells at the edge of the tumor, but they're not getting in. And that's that tumor microenvironment. And that's why the tumor microenvironment is such a big focus of this device and this effort, is to change that microenvironment to enable cells to infiltrate. Those cells with the arrows on the edge and panel 2 are literally knocking at the door. In panel 3, this is what targeted hyperthermia does. It changes that so that the cells have now infiltrated into the tumor. Now, when you look at that, you say, why isn't that just riddled with cells? Well, this is the effect of THT alone. This is an immune infiltrate of natural. This is a natural response to immune infiltrate. And it only takes a small amount of immune cells to affect a massive change, because these cells are not individually. There's enzymes being released from these cells that are causing these cancer cells to die. And so that immunogenic cell death, or that immune cell death, is the effect. A small army achieves a big result. And then, of course, to the right, the 4th panel, what we're seeing is if you take those immune cells and then you fire them up with a Keytruda or a IL-2 or whatever immunotherapy is on your shelf, you can invigorate those cells to recruit more cells into the fight and hopefully have a bigger effect. And so this is exactly what our immune modulation is. We're focused on we're looking at panel 1 and focused on making panel 2 turn into panel 3.
And so really it's the proliferation here that we're looking for, right? How widespread those brown cells effectively are.
Dr. Conrad, I'd be interested in your thoughts on what I just as a pure scientist and a pathologist.
Yeah, I mean, this is the stuff I look at pretty much every day. In the second panel, you can see there's a cluster of cells that are brown, and of course all the purple is just all the nuclei. That's the counter stain that shows where every cell is. And the slightly bigger, more pale cells are the tumor cells that are adjacent to that cluster of brown cells. So panel 2 shows a cluster of T cells, and that's just a little reactive collection of T cells. They're not really doing anything inside the tumor. But if you look at the 3rd panel, there's distribution of the T cells into the tumor tissue. So they're actively moving through it. Like, there's no roadways, there's no blood vessels to take them there. They're actively infiltrating and moving throughout that tumor tissue, which it has to do to mount a response. And then the last panel over, just the difference is even more striking. There's many more T cells that have made their way into that tumor tissue compared to the control on the far left, which is pure tumor and really no T cells in there whatsoever. Yeah, so yeah, I agree. This is showing what we want our immune system to do to fight a cancer is it's being boosted by the THT, and even more so when THT is coupled with IL. Yeah.
Excellent.